everyone. Happy Wednesday afternoon. Welcome to Manufacturing Hub. I suppose if you're listening in podcast format, it's Thursday or later at some point. But for all of our live viewers, happy Wednesday afternoon. Welcome to Manufacturing Hub. We've got Amy on this week. Super happy to go bring her on and talk all about life sciences and a handful of like some really awesome things that she's worked on. For our longtime viewers, you guys will know that we haven't done a ton of deep dive into life sciences. So happy to go ahead and kick that off. I would say beyond that, if you guys are new here, welcome. If you've been here before, welcome back, especially if you're new here and watching on LinkedIn or watching on YouTube. We love to go have fun conversations in the chat. So if you guys are in the chat, come say hi, let us know where you're listening from. Feel free to ask Amy questions. I mean, you guys probably want to ask Amy questions. I imagine that's why you're here. But if you've got questions for Vlad or myself, we're happy to go ahead and ask and answer those questions for you here on the show. Feel free to go ahead and chat kind of in and amongst yourself in that chat as well. We like to have fun, and that is very much the goal of the conversations that we get to have. But without further ado, everyone officially welcome to Manufacturing Hub. This is episode 207. My name is Dave. The guy currently sitting above me is Vlad, and we'd like to introduce Amy Williams as our guest today. Amy, welcome to Manufacturing Hub. Thank you so much for being here.
Awesome. Thank you for that intro. I'm so excited to be here.
Absolutely, Amy. Thank you so much for taking the time once again. Before we dive into the conversation around life sciences, could you give us a little bit of an introduction of yourself, walk us through how you got started in manufacturing, walk us through your career, and ultimately what you are doing today?
Yeah, absolutely. So, hey, everybody, I'm Amy Williams. So currently, I'm a systems architect at Skellig. So Skellig is a life science system integrator. And our mission is to digitally transform the life science industry to help make therapies more accessible. Now, we use the word accessible because it's more than just making things cheaper. Obviously, we want to use technology to bring down the cost of manufacturing because, as we all know, the cost to make anything has to be paid for by somebody. And in America, it's the patient. But there's also more to it than that. There is medicine that's cheap that people can't get due to supply chain issues. And of course, there are diseases we can't treat because R&D costs are too high. So I'll admit, those are tricky problems that I can't claim I've solved all of them yet, right? But as a company, that's where we're going and that's what we're trying to work on now. So what we do today is we help implement solutions and architectures that we think are going to help support that goal. So I'm a fan of the UNS approach. So we do architecture designs that are open, edge-driven. We do automation and MES projects that kind of fit into that as well. And that's kind of what we're doing now. But my background is right after I graduated from NC State University, go Wolfpack, I was an automation engineer in the life sciences as a systems integrator. And then eventually I actually got hired to work at Skellig to help with a really cool project, which I hope we get to talk about, which is Operation Warp Speed.
Mm-hmm. And I guess just to double down or ask you a little bit more about your background, what was the program that you studied at the university?
So I did actually biomolecular chemical engineering. So it was chemical engineering, but with a focus on biotechnology. So stuff like bioreactors and like gene editing and kind of like the science behind it. It's funny, it's actually really common in the life sciences for them to hire chemical engineers to help with the automation. And I think that's because like a lot of the automation we do is from a code standpoint, kind of simple and straightforward, but the hard part is understanding how to actually support the process and being able to talk that lingo with the process engineers and with the operators.
And that's super interesting, right? Because the ongoing, I would say, debate, I want to say in our industry in a general sense, right? Do you hire someone with a process background and teach them controls and automation and what it takes to extract and work with data, let's say at the SCADA and MES layers? Or do you bring in, really good software engineers and maybe teach them the process, the manufacturing, and what comes with it? And that's why I think your background was very unique in that sense that you were able to understand a process unlike, again, a software and electrical engineer could, and then you could just fill in the gaps on the automation side. And if I may ask you maybe to expand on that a little bit more, what was that learning curve like, right, for someone who's maybe looking into a career in automation or in manufacturing with a I want to say like more life sciences type of a background? What can they expect out of it?
Yeah, that's a really good question. So I would say it's kind of a double-edged sword. It depends where you're going to go in the software stack. So I think if you're focusing more on the automation side, I've always heard the phrase "KISS, keep it simple, stupid."
Mhm.
So I think maybe if you're really good, I don't know, but maybe if you're extremely good at like software and advanced like coding, you might get frustrated with how maybe basic your code is going to be on the plant floor. But again, it's because it's supposed to be a simple thing that just works all the time and is easy to maintain. And I only say that because that's kind of the vibe I get when I talk to people who, for example, are really into OPCUA and they understand that so well and they're brilliant and they're intelligent and that's wonderful. But sometimes what I hear from them, I'm like, that doesn't map on to what I actually need to do today in operations. I want it to be simple. I want it to just work right out of the box. Um, so I feel like the learning curve isn't that bad as long as you keep an open mind and you realize that it's kind of a different world. Like I don't know what's your thought? Like, do you think you can learn automation best practices online? I don't know if they have like a code academy for PLC coding or
Hmm. That's an interesting question. I guess, look, for me, what it felt for me is that obviously I had gaps on the process side, right? So I understood electrical systems really well because that's what I spent a lot of years doing in the lab, and learning through measuring things using an oscilloscope. So I would say that I had a lot of intuition on when, let's say, systems failed and how to troubleshoot systems. But on the process side, of course, transforming raw materials into finished goods, even in food and beverage industry, yes, I understand how ingredients get added, but I wouldn't necessarily easily understand the chemical processes that happen under the surface, if that makes sense, right? And so a
right.
lot of times for me, it boils down to someone needs to explain to me what is expected out of a process for me to build, let's say, a control system or extract data. But I would say, I probably would never be as good as someone in food sciences or life sciences that has that background, right? And maybe they will not have the same intuitions as me when it comes to electricity per se. Does that make sense, right?
Yeah, definitely.
That's
No,
kind of how it
absolutely.
felt with my. Yeah.
Yeah, no, I think that's a really good idea. And probably the best answer is it's not either or, it's and. Like, you probably want to
Mhm.
have people with different backgrounds so they can play to each other's strengths and weaknesses. Yeah.
Let me ask you maybe like a follow up, but also a fundamental question, right? So, how do you define life sciences, right? So, I've spent a lot of years in FDA regulated environments, but life sciences is not something I would have come across too often. And I would say also maybe the viewers or the listeners would probably not all be familiar with what that looks like.
Yeah. Okay. So that's a good question because honestly, life sciences is kind of an umbrella term. I think officially it's like technically work is centered around research and development that's focused on living things. But if I want to be practical, I would honestly classify it into three buckets if we're talking about the life science industry, especially like the US. So the first one would be pharmaceuticals. So that's if you're doing chemical synthesis to make a medicine to treat diseases. And we commonly refer to that as like small molecule. Now, like a pop culture example, I would say, is that crystal clear blue meth made by Walter White. Because technically speaking, that was a small molecule. But in the life science industry, we actually call pharmaceuticals more of like pain relievers and traditional medicines. The second bucket I would say is biotechnology, where you're actually using living organisms to manufacture products. So unlike Walter White, he was just mixing chemicals in an RV. Instead, you're probably going to use something like a bioreactor to grow cells that produce your therapy. Now, technically speaking, one of the earliest examples of biotech would be in food and bev, which is beer. Fermentation is using yeast to convert sugars into alcohol and CO2. I suppose the big difference, though, is that when we say life sciences, we're normally thinking more of these like medical approaches like vaccines, gene therapy, and biologic drugs. And then the third big bucket that I would say is life sciences, is medical devices. So that's making instruments to diagnose, monitor, treat medical conditions. And that can range all the way from like a Band-Aid to something as intensive as a pacemaker. Now, I'll admit I haven't worked much in that industry, but then kind of supporting these three are also CMOs, which are contract manufacturing organizations. So they don't necessarily own the intellectual property, but they support the testing, the refining, or the manufacturing of drugs or medical devices.
Interesting. So, but ultimately, this falls still under the purview for the most part of the FDA, right? Because all of these, I guess, well, to some extent, not the devices themselves, but they still come in contact with, your skin, the body, et cetera, et cetera. So, I'm assuming the FDA regulates a lot of what happens with
Yeah,
these. Okay, gotcha.
absolutely. And when people are looking to hire in life sciences, usually that's like a big criteria. Do you have what's known as like GMP experience, which is essentially documenting everything, signing your solo on all the documents, understanding how to act in an environment that can be audited by the FDA.
Interesting. So I guess it's a matter of definition. In that case, I guess I have been part of life sciences projects more than I thought, because ultimately fermentation processes, as you've mentioned, are very common in food and bev, and there's a lot of, I want to say, that type of transformation even with milk, cheese, and a lot of like dairy and farm products. So very interesting. Appreciate that breakdown. Dave, what are your thoughts?
Well, I think first I'd like to commend Amy on her pop culture reference. This might be the second time Breaking Bad was referenced on the show, but it's definitely the first time meth was used as an example. So I like to commend Amy for that. I feel like Vlad, you kind of dropped the ball on calling out that fantastic example that I imagine all of
It
our
was really good. It
listeners.
was really good.
Yes. That is good. I really appreciate that. I really appreciate that, Amy, and I really appreciate kind of the descriptions of the three buckets that are life sciences. And so you talked about I think you said GPO, the documentation, the kind of what the FDA process looks like. Would you say, in your experience and talking to other people, that's kind of the big differentiator on life sciences processes and on life sciences processes and perhaps other industry processes, is all of the documentation and all of the certifications that one has to go through?
Definitely. And I think there's probably a good caveat worth putting before that, is that one of the things about most life sciences is you can't just manufacture something because you know how to. You can only manufacture something if the FDA gives you approvals. So I think in like the medical device industry, you can classify them differently. So if it's like a Band-Aid, way less stuff you have to submit. But if it's a pacemaker, you might need clinical trials. And I know for a fact that at least in biotechnology, and I think pharmaceuticals as well, you both need to go through clinical trials before you can manufacture something. And those are time consuming, those are challenging, and those are very expensive. So I guess, and I want to mention them because it kind of ties into kind of how the industry works. So one thing I want to talk about is that these clinical trials, they're usually done by an independent third party, but they're funded by the company and they're required and they do take a while. So there's usually three phases. The first one is about safety and dosage. It's small. It's like 10 to 100 people. It's a few months. The second one is 100 to 300 people, could be months to years. And again, you're starting to check more of efficacy and side effects. And then phase 3 is like thousands of participants. That could take 1 to 4 years. And you're confirming the efficacy at scale. And the thing is, I love asking this question, like take a guess of what percentage of new therapies make it through clinical trials.
Ooh,
My guess would be
all the way through
10%.
all
All
three.
the way
Yeah.
through phase 3. Take a guess. What percent?
I'll guess 10%.
Yeah, I was gonna guess 5 to 10%.
All right, you guys are good. That's the winners, right? So we're talking 90% failure rate.
Yeah.
And if something fails, you want it to fail early because it's expensive. Like phase 3 alone is hundreds of millions of dollars because you're monitoring thousands of people. And like, there are some guesses that bringing an entire new drug to market can cost like $1 billion because you're counting for the fact that 90% fail. So that's another big distinguisher of the life sciences, is that these companies have to be able to offset the insane amount of cost that they invest up front, even before they start to get into their manufacturing.
And just so, if I can ask a clarification question, just so I understand, in these three phases, I'm assuming you're not going to start manufacturing until you get a lot of certainty and you're a bit further along in the process. So you're doing small scale trials of creating whatever product at first, and you're at the same time ramping up maybe a production line. How does that maybe timeline work as you're going through the phases?
Right. So because it's so expensive, traditionally with most medicines, manufacturers are not going to start doing like large scale manufacturing until they're in like phase 3 clinical trials, and if it looks really promising. But normally, yeah, they're waiting till the end of that clinical trial stage because it's a lot of money to invest into manufacturing a product. Yeah.
So they don't really know, I guess, based on the data, even in phase 2, what the certainty is of getting through phase 3, right? They kind of
Yes.
know, but it's a lot of risk and uncertainty still at that time to invest. Very interesting.
Yeah, absolutely. And you see it all in the stock market, like, oh, we made it through
Yeah.
phase 2. Great. Oh no, unpromising result. So
Absolutely. That would be a roller coaster of emotions, especially for the scientists working on those particular drugs. Because I have to imagine you only get so many cracks at the opportunity to create a new drug unless you are leading the research facility and then you've got a dozen drugs under you. So you only have so many opportunities during the career. So that would be interesting. I wanna go talk about Operation Warp Speed, because I feel like we've set everyone up for what normal processes look like, and now we're probably going to go blow those timelines out of the water. So, Amy, for the listeners at home, and probably for background on Vlad and I, can you tell us a little bit about what Operation Warp Speed was, and how you were involved in that, and then we'd kind of like to dive into some of those learnings, and how it was different than what we just talked about.
absolutely. So I feel like to properly understand and kind of appreciate Operation Warp Speed, I kind of want to tell like a little bit of a story. So if you'll bear with me, because the thing is, like, COVID today, thankfully, is not a very scary disease, right? Most of us have some kind of immunity, and we have a variety of treatments that can, help you in case you do get it. But again, that wasn't the case 5 years ago. So let's all hop in my time machine and we'll jump back. So it was late 2019 that people in Wuhan first had symptoms, and China notified the WHO in early January, and it quickly escalated to full lockdowns of the city. Now, this was scary because it was fast, but it was also far away, right? There was hope it could be contained in China. Well, that hope was quickly crushed because in February, it was when Italy's hospitals started getting really full. They locked down their country, and there were these like sensational headlines that the healthcare system was on the brink of collapse. Now, normally I don't like when the media makes these crazy headlines, but this time I think they were right, because this was horrifying. This was concrete evidence that it was more than a regular flu, because it was something that could actually take down a system. And if a hospital fails, it doesn't just hurt the COVID patients, it hurts the healthy people, right? Because if you get in an accident, if you have a heart attack or a stroke, you're going to be impacted because of the lack of resources. So at the same time in February, we're starting to see community spread of COVID in the US. March 13th, the US declares national emergency and states start locking down. But here's the problem. Lockdowns kind of work, but they also kind of hurt the economy. It was like 6 weeks later, you had 26 million people had filed for unemployment. So the US was stuck between a rock and a hard place. You could do nothing, which risks the health of a lot of your population, or you could try to lock down for as long as you can, but that's really going to crush the economy, which is also devastating. So everybody agreed those weren't good options. What we really want is a vaccine to protect our vulnerable and allow us to open up the economy again. But here's the thing. Remember how I talked about like those clinical trials and all that, right? Traditionally, it takes about 10 years to bring a vaccine to market. And we didn't have that time. So this is where Operation Warp Speed came into play. It was launched, I think, officially April 30th by the Trump administration with a goal to bring a safe vaccine to market by 2021. which raises the question, how did they do it so fast? Well, the main reason that vaccines and in general a lot of therapies take so long is because they do steps in sequential order, like Vlad pointed out. So first, because it's so expensive, they need to get investor funding. Because as we said before, it could be millions, if not billions of dollars. Then you have to do clinical trials, stages 1 through 3. And then after clinical trials, they submit all that data to the FDA, who then takes, a few months or so to review it and approve. And then only after it's reviewed, they can start manufacturing. And then if it's something novel, that means you're probably going to build a new site, which can easily take a few years. So during Warp Speed, instead, they did everything in parallel. And the government provided a lot of funding, and it didn't bank on just one vaccine. So to start out, it was like about $10 billion were allotted to give funding to 6 different life science companies to produce a COVID vaccine. Now, dozens of companies applied for funding, but they specifically selected for ones that had promising phase 1 clinical trial data or animal data and could meet the tight timeline, which meant they're not going to build a new facility. Instead, they're going to repurpose what they had. So by providing the funding, they eliminated the financial risk and time it would normally take to get the private sector to give investments. And second, they did the manufacturing and clinical trials and approvals all in parallel. Now, this was a huge risk because as we talked before, most things don't make it through clinical trials. So normally the private industry doesn't do this because it's really risky, but this was extreme circumstances and the government was helping with some funding, so that allowed the private sector to take that risk. And the third thing that they did was rolling approvals, which meant the FDA could make approvals for emergency use authorization based on the available data rather than the full biological license approval. And so what was cool was Skellig got hired to help with the automation for one of these sites, and then they hired me to help go on site to help with all of the commissioning and support. I have to say, it was really surreal going on site because at the beginning of the pandemic, there were strict lockdowns, but I'm used to being the consultant who gets banished to the trailers in the parking lot. But for this project, everybody was sent to work from home because the last thing you want is the people helping make a vaccine to stop because they got that disease. So when I went on site, it was great. I had the closest parking to the door. I'm walking through this building. I probably had an official seat somewhere, but it was honestly a free-for-all. Like those fancy glass offices that normally have to book a week out for possibly an hour, you could live in there. No one would really notice. So it was a really, really cool experience. And I feel like, unless you want me to stop, I feel like the best way to explain the project is kind of to talk about it in 2 parts. First, from a process perspective, and then kind of second, what's that actually look like behind the scenes?
No, it's super interesting, right? Because I think a lot of us have read as to what happened, right? But firsthand experience is definitely a lot more interesting to learn about. And again, especially in manufacturing, right? Like again, I've went through the shutdowns, and I was going to food and beverage facilities who were just losing people, and the concept there was just to make sure everything's automated so that we can produce more, but it was not the same challenge at all that you're describing, right? So that's why definitely appreciate the breakdown. So really curious to hear more.
Okay, awesome.
Yeah.
So the first thing that I want to talk about is the fun part, which is kind of like the process side. What were we trying to do? Now, it's a little tricky with NDAs to talk about too many specifics, but I'm happy. I found a public press release from the site I was working at in 2022, where they said themselves that they were making a recombinant COVID vaccine using baculovirus. That probably sounds like a bunch of confusing words, so let me break that down. Mm-hmm. A recombinant vaccine is one that uses another organism to make the antigen. And a baculovirus is one that can only infect invertebrates. So that's animals who don't have backbones. That's not you or me. That's more like worms or arthropods or jellyfish. So what the scientists do is they genetically modify that baculovirus so that it's not going to tell the host cell to replicate more of the virus, but instead to make more of the desired antigen. So in this case, it was part of the COVID spike protein. And the antigen is what triggers an immune response in a person. So that's cool because it's safe, right? It can't infect people, and it can only tell cells to make a piece of the protein, of the spike protein. So for this process, they had non-human vertebrate cells that they're growing, usually in a bioreactor, and they're feeding them food, which we call media. Now, usually you try to grow the quantity of these cells, so as many as you can. So you're scaling up these cells into bigger and bigger vessels to try to get the biggest batch that is stable. Then once those cells reach a certain density, they are then infected with this baculovirus, where now these cells are producing the desired antigen. And you then have to wait a little bit more time for enough of this antigen to be produced. So this is a slow process. This doesn't take a matter of minutes. We're talking like many, many days to get to this point. But now you are left with kind of a biological soup. It has the cells, it has this virus, and it has your therapy, your antigen. And that kind of brings us to the second half of the process, which is purification. So basically, you take the contents of your bioreactor and you start running it through lots of different filters. Now, I won't say specifics on which filters we use, but like a few common things that you're filtering on are ones that work based on size of the particles, ones that work based on the charge. So for example, ion exchange, in a liquid, everything usually has a positive or negative charge. So an ion exchange filter might have a resin or membrane with the opposite charge to attract that particle. So it'll bind it, it'll hold on, all your waste flows through. And then at the end, you can change the pH or salt to elute what you captured and continue on with filtering. They also have like affinity-based filters are common in life sciences, and that's kind of built based on a biological chemical recognition. So it's like lock and key. It's like it has a binding site designed to stick to one molecule. So it's all about like, does it shape, fit? And, you know, I think frequency tests are actually affinity based because they bind to HCG. So that's the second half, and it is much quicker than the first because you're not waiting on cells to grow. And of course, all of this is happening in a clean environment. Now, when I joined, we were just installing equipment still. We were doing water runs, so we got to run around a bit with only the hard hats and the safety glasses. But of course, as the project progressed, eventually everything was elevated to a GMP clean room status. So that means everything is strictly temperature and humidity controlled. There are airlocks between the hallways and the suites, and they have strict rules on how clean the air is. And I mean, they're measuring this in like parts per cubic foot. And of course, we had to break out what we referred to as the bunny suits. So if you want to go on the floor, first you gotta go in the locker room, take off your day clothes, put on these scrubs. So those are like sealed in a plastic bag. Then you put on the booties, the shoe covers, the gloves, the hair net, probably beard net for some of you, and glasses. And if you wanted to bring in like a company provided phone, that also has to be wiped down with IPA and bagged. And then to go into a room, you're putting on another layer of gowning, some more jackets and more booties. Now, I'm pointing that out because that was and is a challenge with the life sciences. There is no running out to the plant floor. Like if you need help and want somebody physically there, well, I hope you have 20 minutes because there's nothing fast about this gowning process. But that was kind of like the glamorous side, the like fun process side of life sciences, but it wouldn't be the whole story unless we also talked about all the regulation impacts of how that impacted like the project execution. So everything I'm saying now is kind of standard across life sciences and not just warp speed, but kind of just impacts the industry as a whole. So the starting point of most projects is a master validation plan. So it's like a roadmap. It's an overarching document that outlines all of the sub-documentation and processes. So here you're going to say, okay, equipment will be brought in following this SOP for validation. Automation will be validated following this SOP for validation. It justifies the overall project strategy and scope. It usually says you're going to do the 3 big validation elements of IQ, OQ, PQ. So that's installation qualification, verify it was installed correctly. Operational qualification, does the equipment run like you said it would? And process qualification, does your process actually make what it's supposed to in a repeatable manner? Now, I know every industry follows the same logical sequence, right? Everybody brings in equipment, fixes it, tests it, and then starts production. But difference is for life sciences, that's not very fluid. Every stage is defined upfront in writing, and there is always a clear barrier between each phase. And that's usually tons of signatures and document approvals to move forward because that's required from the FDA. In fact, it's pretty common to have an entire role called a CQV engineer. So that stands for commissioning, qualification, and validation engineer, whose job is to support documentation and validation for all these stages. So they're an independent group. They're separate from the engineering team. And they write test protocols that kind of define each stage, and then they execute them and get the approvals to move on to the next one. And so I mentioned that Because in life sciences, you have to write and approve everything before you can actually do it. And the result of that is decisions get made early. And so you tend to get stuck with them because you had so much documentation saying, hey, this is what we're going to do. And like one big example of that is system architecture. That's a big decision usually made pretty early. And that's because you can't just one day say, I'm just going to start using a new platform. Now, there's like an entire system validation plan that you have to write and execute to prove it's viable for life sciences, right? So that plan will include requirements for 21 CFR Part 11, which is, do you have user management? Can you ensure the data is only accessible to certain people? It'll have a risk assessment deciding what criteria are needed. Are audit trails applicable? Can you do redundancies or disaster recovery, right? And this is all because the FDA requires full traceability. And it's not the FDA's job to be the SME in every process and software on the face of the planet. So you need to have this document showing what you're doing and how it ties and meets the regulations provided by the FDA, kind of translating it into a language that they can understand. And if you're in a full GMP environment, this is pretty time consuming. So like maybe in other industries, if you want to change a parameter, right, you can log in and push the buttons, change 5 to 6, save, maybe download, and you'll be done. And that's a matter of minutes. But for most projects, especially ones from like 4 or 5 years ago, doing that is probably going to take like 3 days because you're going to have to open the change control. You're going to have to do testing, get it released into production. And so it's kind of an ongoing challenge for life sciences to move quickly. Now, knowing all of that, the project I was on did manage to move quickly. And I kind of want to talk a little bit about how they did that. So first, go ahead.
I was going to say, Amy, I think that this is fantastic. Before we go talk about your project, maybe like a couple of clarifying questions. I know I have a couple. I'm sure Vlad has 10,000. But I will go start because I so rudely interrupted you before we go talk about your project in particular. So when you were talking about adding the media and growing the what's the correct word for the combined media and everything that you're growing? Is there a technical word for that process?
I bet you there is. I just call it growing the cells. Like that's your media.
Perfect. So on the growing the cells part, I imagine it started by someone in a laboratory environment so that they can figure out how much of the size of the cells and media that they can grow in this process, and then it's your job or was your job to go and automate that process. So like is there starter cells that they put in and starter media? I'm interested to know how that goes, because you were describing that and I'm like, I can see a person in the lab doing it, and you're like, and then it's perfect, and then we just go filter it all away. So I guess a couple of clarifying questions on that, if you wouldn't mind.
Absolutely. So that's a really good point that I probably should have mentioned. You're spot on. So most life science pharma companies, they do have an entire lab space. And actually, I love the lab spaces because usually that's somebody's full-time job to figure out how do we scale this up. So some scientists and some R&D place is probably working with like the cutest little, I think a popular one, if you want to Google, is like an Amber system. They're these teeny little tiny bioreactors that are like, I don't know, 10 inches tall, 5 inches wide. And they're just figuring out how to make the product. But then it'll go to these like lab areas where people are, again, really smart scientists trying to scale it up. And those areas are actually a little bit less GMP because they're not making product that goes into an actual person. They're just trying to figure out how the heck do we get this thing to work in a big manufacturing environment. So that, luckily, when I came in, somebody, a very smart person, already said, okay, I understand that you're actually going to start off with a flask. You're right. You always start with a small scale of like a flask of cells, but then you go through the process of expansion. So you start with that little flask, who then puts it in maybe like a 50-liter little wave bioreactor, and then you can scale it up to maybe 100-liter, and then 200, 500, 1,000, 2,000. I mean, you can scale up as much as you like. But all of that equipment is, at least when I joined, was predefined, and then it was my job to help with the automation control to make sure that we are meeting the very, very tight process control parameters that they told us is what we need to do to get these cells to happily grow as we transfer them from small vessels into bigger vessels to eventually introduce, that virus and filter it out.
Okay.
If I can ask a clarification question, I guess along the similar lines, right? So when it comes to instrumentation for that kind of process, right? So you mentioned obviously there's temperature control, there's probably pressure control, there's level control, right, for the tanks. But I'm assuming it's very difficult to detect at the microbe level, or I guess at the cell level, what transformation actually happens. So a lot of that gets brought back, I'm assuming, at a what's it called, at a statistical level, and tested in the lab in order to see like what's happening within the process at different
Yeah.
stages. Okay.
Yes, absolutely. So that's a very good point, and that's definitely a challenge, is that like almost at every stage of this process, you have to have people come out and take samples who are then going to bring it back to the lab. And that's hard because now you're introducing this manual component of somebody extracting, a sample from a bioreactor, and that can lead to all sorts of problems, right? Like make sure that they open the valve, close the valve the right way, they don't actually trip on something. And this is something where I kind of wish that kind of ties into industry 4. I mean, we can talk about more later, but I'll mention it now. One thing that would be great is if we could get more sensors on the equipment, like process analytical technology, or even more like video information, just because you can't get around that. You have to take samples, you have to, and what's really common in life sciences is also single-use equipment. So all of that is like plastic. You have to set it up every single time. So again, it's great because single use is typically faster because you don't have to clean it and it's more sterile. But it does introduce this complexity of, operators are going to be handling this. I don't think that's going to go away anytime soon. And it does introduce this uncertainty, this room for error if something goes wrong. And it's a little hard because if you just remote into like your PLC level control, you miss all of that added context.
Right. Yeah, that makes sense. The other question I had, right, and maybe this is a good transition to the project you worked on, right, is I'm assuming a lot of these projects are not necessarily greenfield, right? You're not starting from complete scratch at most of these companies. You already have some kind of a working line or maybe something that's already in place that you can draw from, right? You're not starting from a completely open facility. And even if you do, it's just to some extent a copy paste maybe of something that already exists. Like, how does that work, and how different is it from, they're making this type of a vaccine, now we're going to this type of a vaccine? Is it drastically different, or is it just a like minor adjustment of the process? What does that look like?
Mm-hmm. Yeah, no, that's a really good question. So first, for the project that I was on for Operation Warp Speed, that's exactly what they did. They said, and this actually helped them get to market faster. They said, okay, we already have a vaccine process that uses most of the things that we're talking about, and it's validated and it's safe. So we're going to repurpose this and try to just tweak what we can to make it work for this project. I worked at another site, though, that dealt with manufacturing a lot of different things with the same equipment, and that was kind of called changeover. So most of the time, a site is trying to reuse the same big pieces of equipment because you're not going to casually wheel in and out like a big stainless steel bioreactor. However, if you're making a new therapy, it isn't that uncommon. I'd say more so in the downstream side that you have to bring in some kind of different filtration skid for that. So usually what happens is, depending on, I guess, the type of site, but usually control strategies are in place where hopefully you can change a lot of the things that are different, which are going to be like your DO, temp, pH, that can be driven by parameters. And so hopefully if you're going to make a new product, the actual control logic doesn't change that much. That's always wishful thinking. You can't get around it. There is going to be some process change, but they do traditionally try to not, there are a lot of sites who do try to make different things within the same like equipment train, but it is a bit challenging. So one thing that's really challenging about that is the GMP aspect of adding code to the plant floor. So this is actually a totally different site I worked at, but we were doing exactly what you said. We were going to make a new product with the same existing equipment. So we just needed to add new code to the stuff on the plant floor. The hard thing is, though, there are lots of rules about updating the code on the plant floor. So something that you have to do is you can only download an update if that piece of equipment is not running. Now, not only do you have to do the download, but you also have to release it, which means you then need to get the signatures of like the process people and the validation people saying, yes, we verified that their testing was good. We verified that this is now officially released. And so the tricky thing is, especially like 4 or 5 years ago, is a lot of life science companies would have schedules literally in Excel, or it'd be an email you get like in the morning. This is what we're running today. And so as the automation engineer, you're looking at that and you're like, okay, I may or may not have a 2-hour window at 3 where I can do this download and get this group of people to sign this piece of paper. But I don't know, because it's, you know, it's a piece of paper, right? Like I'm going to have to log in and kind of like stalk the batch the whole day and get a meeting set so we can get all of this signed and released in time. And so that's definitely a gap that I think we can gain a lot of ground on, that maybe people who do capital projects don't realize because they're not doing the day-to-day change controls. But there's a lot of benefits if you can get integrated systems, especially scheduling, and get that data visible just to help with like day-to-day operations of doing updates.
Got it. Dave?
Interesting. I'd love to talk a little bit about control systems, but maybe before we talk about what's currently being used in life sciences, we did interrupt you kind of mid-story of operational
Oh, yeah.
updates. So can we let you go back to the second half of that story, and then we'll go talk some more specifics, please?
Absolutely. Okay, so for the project I was on, it did move fast. And so I do want to shout out this. The main reason it did was because they hired the right people. Honestly, it was an aggressive project. It wasn't easy. You know, if you're working late, when you're working on weekends, having the right morale goes a long way, because especially like things go wrong, it's inevitable. It's good to have the culture of solving problems and not just like pointing fingers. So utmost respect, great people will probably factor your project success more than you realize. The other thing that was really cool was this was actually the first time I'd ever seen single-use equipment. Again, this was like 4 years ago, but it was really, really cool. So as I kind of briefly mentioned before, single use is not like what you see in like typical breweries where you have like the big stainless steel tanks. No, single use is literally just plastic. You use it once and then you dispose of it. And the advantage of that is you don't have to worry about cleaning, because cleaning involves a lot of water. If you're doing like clean in place or steam in place, it's very time consuming, it's very expensive, and it increases a lot of risk for like bioburden and contamination. And now third, this is going to sound crazy and maybe you're going to disagree. But something that this project did that made them fast up front was they actually used a lot of paper. Now let me explain why. The control system that they were using has a MES system with it that is kind of rigid. And this was a novel process. So we were still figuring things out at the very beginning stages. We didn't really know exactly what the operators had to do. There was a lot of going back and forth and figuring out what the operation was. So what they did was, in the beginning, it was paper-based just to allow for that agility. And then later, as the process kind of got ironed out and figured out, then they started introducing like this digital MES system that was a bit more rigid and kind of harder to update.
So who was I guess a couple of questions just to clarify in I guess in the same order that you mentioned. So the single use, those are plastic vessels, as I'm assuming, for the bioreactor, or is that something else? Like, am I just picturing like a massive plastic tank that gets thrown away after the batch? Like, is that right, or is it?
Absolutely. So it kind of looks, so there's usually like a metal like jacket,
Okay.
and then there's a plastic bag that goes inside. And then you have single-use probes as well. Well, yeah, that you can like plug in. I mean, part of it is single-use. The part that's contact with the cells is single-use. Yeah, so that's for the bioreactors. We even had, I think, single-use filtration skids. We had single-use columns. So all of that was really novel. Because the cool thing is, if you're not using single-use, I kind of mentioned it, for the upstream, you have to clean it. And then for the downstream, if you're using a column and you have to pack that manually, because part of a chromatography column is like filling it with like the resin or whatever the membrane is, like that's a manual step that again takes a lot of time. And it's super fast to instead just wheel this thing out that you use once and then throw out. But it is expensive. But people have done the math. Apparently, it's not as expensive as actually owning some of these things.
Interesting. But so even the
But that also
oh,
kind of
go ahead. Sorry.
no, I was gonna say that also. Okay, I'll talk. Brings me to another thing that they did as well. They also leveraged a lot of like vendor skids where they could, especially with like some of the downstream things, which meant that when it was delivered, it came with a documentation package. So you didn't have to spend time like sitting there yourself going through the hassle of like, okay, I'm gonna do all my tests and stuff. It was already like GMP out of the box more or less, and then you just needed to do like wrapper protocols to get it in.
Mm-hmm.
That definitely cut down the time up front. Like there's no denying that. Like those things got into the suites fast. But at the same time, you also kind of sort of pay for that later, because if you needed a code change, what do? You have to call a vendor, ring them up and say, okay, I want to change this. You need to come out here, and you're kind of beholden to their timelines. So that's kind of an interesting double-edged sword about this project. They did a lot of things to get things fast up front, but I will say in the long run, you also saw some downsides of it. Because especially with the paper MES, it's really hard to do troubleshooting if the information of who did what is sitting on a logbook on somebody's desk. I can't, like, I'm stuck setting a meeting waiting for somebody to go grab that and like read it to me, you know?
Yeah, the question on the paper MES side, right? And it's not that I disagree. Obviously, I don't have full context of what events actually took place to get there. But just so I understand correctly, I would assume they're logging some metrics from the line in a manual fashion. Like someone actually sits there and writes down whatever they're measuring on the quality side, whatever they're measuring on the time side, and then someone collects that and then presents you, hey, here's the maybe like the timesheet or the report of this specific batch, and then you get a bunch of those over a couple of shifts or a couple of weeks, and then you can make decisions. Or what did it actually look like to have a paper based like SCADA or MES?
Yeah, so the interesting thing is they actually used a DCS system. So that kind of crept a little more, maybe it did more than just PLC. So we did have automated like digital prompting saying like, okay, add sample here following your SOP, which may be a paper thing. So this is also something else that's interesting about life sciences, at least from a couple of years ago. Everything, probably because of the fact that you have such strict user management, things are pretty siloed. So as an automation engineer back then, I can't tell you really what happened. I know for a fact that they had to write down in their paper logbooks a lot of information, but if it wasn't being tracked in my DCS system, I knew it was being tracked somewhere. But then all of that somehow got reconciled with quality or with the MES team or somebody to eventually make that giant batch report that then gets submitted to the FDA. Again, for this project, I was just an automation person who was there early, just doing the DCS control. And so I know for a fact that like we would have some areas where if they had to set up the bioreactor, for example, we would have to code an initialization phase. And in that initialization phase, you're saying like, okay, well, now that you have a bag there, we're going to just do a pressure test to inflate it and continue with manual operations in your SOP. And I don't really know what those were, right? Those digital work instructions were things that were very paper oriented.
Let me ask you a question. So, Amy, the value of the paper MES is we can write down whatever information we want, and it doesn't have to be within this GMP certified, validated solution. And so,
no,
okay.
It does.
Okay.
It does, but it's really fast it's not fast. It's faster to update.
Okay.
Because what are you going to do? Maybe you take some notes and you go back and you have a change control where you redline it, you print out the next one, and you go to your next batch. I don't want to talk poorly of this MES system, but there are some MES systems out there that do not do very well with change. And so when you start to introduce this whole change control process, because it's like, it's very similar to DCS. If you want to make a slight modification to your MES recipe, well, you don't just make the update. You now have to go ahead and make a test protocol that has to get pre-approved by like your quality person, maybe the engineering group, maybe this computer validation group. Then you have to make your change. Then you have to get another MES person to review your change. Then you have to get permission to like import that into production. It's like there are so many steps the moment you introduce automation. that if you're going to be making a lot of changes because you don't know the process, they add up very
Okay.
fast and can cause a lot of delays. So the idea was, yeah, no, this is still regulated. We still need to have change controls and stuff, but maybe it's easier to figure out what we need before we actually like kind of hard code this. Now,
Okay.
what's cool is I'm talking about an MES system for 5 years ago. If I'm going to talk about, like, for example, like Tulip is really quick to configure, I think that maybe if I was in a similar project again, I would say, okay, we're actually going to build small little apps that are mobile. They aren't super dependent on each other, and you can actually make quicker changes, and it's not such a heavy burden.
Okay.
But I don't know. Honestly, I'd like to hear your opinion. I think it's a kind of a debate in the industry right now about if that's the best strategy.
I think it's interesting. So my background is in aerospace and aviation with the FAA. Not all that dissimilar, except the rules are like 50 years older, basically, right? So the Bible, the FARs, is written in the 70s or 80s, and maybe hasn't been updated since the last time, somewhere in there. So those are all much older and very much rigid to kind of this point. I suppose that the concept of going and mapping out and understanding what you need before you spend the time and money going and coding and building that, it makes sense if you were to build it for the first time, but I would imagine, and Amy, you tell me, I would imagine, I guess in my experience of building MES solutions, you get to the point of we've done it once or twice or 5 times, and we've got a pretty solid like 80, 95%. This is what it's going to be. We might change or add some sensors. We might change or add some other things, but you have a fairly good idea of what the solution is going to need to be at the end, and you just have to go plug and connect those dots to specific machines or specific sensors, or this is going to be disposable, or we're going to repack this, and these are the data points we need from that. That's my thought after having 7 minutes of paper MES conversation. So I assume that the chat is absolutely going to flame me, because I imagine that I'm missing some huge.
I'm not pro paper, okay?
I imagine that I'm missing some huge value. Again, as you said it, my jaw is dropped. Like, this is what I've spent the last decade of my life trying to fight against. This paper MES that is just like, paper notes written and set down on one person's desk. And if I need information from 10 years ago, I have to dig in that stack because it hasn't moved in a decade.
Oh, no, okay. Let me protect my reputation before I lose my job. Like, I'm not for paper MES either. No, it causes so many problems. It was a nightmare when I was on call because I had no idea who did what. I don't want the direction. I don't want that to be the way the industry is going. But I guess I'm explaining it was like that for those reasons of these like giant systems were hard to maintain. I'm 100% with you. You should 100% digitize your MES. Please, everyone in life sciences, do that. But maybe there is a strategic case to be made that, like, it is hard when you're scaling something up for the first time to be flexible. So yeah, maybe iron out that process first, but then, oh my gosh, please digitize that.
Yeah, I would say, if I may, on my side, right, like having worked in a very regulated environment for Procter and Gamble, where it was very much like you've described, I think what you've described is it was very structured, right? And so let's say I had tasks where I needed to align a test in, like, let's say 2 weeks to 4 weeks, and that was scheduled, a very tight block in between batches, and that was kind of the norm, right? Like, it was expected that, hey, if we want to make some change, Vlad and whoever is on the process side are going to go conduct this validation. They're going to confirm the code. Vlad is going to sign off on the code. This process guy is going to sign off on the quality side. We're going to pull some samples. But then you take that, right, and that's the process, and you have, let's say, a 1-year runway of projects, and you're like, hey, let's do that in 2 months, right? And so obviously at that point, I couldn't tell you what would be a better approach, right? Because at that point, everything kind of gets thrown out the window, and we're just looking for pure efficiency, and we're trying to not necessarily cut corners, right? But we're cutting as much bloat as we can, which ultimately I could see why it leads to some processes needed to go back to more manual, right? Because simply the systems are not designed to track in a similar way. And once you, I guess, get out of that crunch, then you can resume sort of normal operation. So I understand, I guess, like why it was done that way, because you need the information. At the end of the day, if it takes your MES system, whatever, to process that information, you don't get it in time, then you're just going to be slower for whatever reason, right? And if that same process applies, they need to check, they need to double check, and it's better for you to get a paper report, within a few minutes than to wait on whomever to release it. So
Right. And this was specifically like trying to get that vaccine out the
yeah,
doors, like getting it to valid as fast as
yeah,
possible. What's nice is we usually are not in pandemic states. So for the 100th time, I'd be clear, life science industry. I definitely think that if you do have the time, yeah, iron it out, but then definitely make plans to digitize your MES system. And not only just digitize it, but make that data accessible, because you might not realize how many people, again, on your day to day benefit from just knowing who is doing what, especially if they're trying to troubleshoot.
Absolutely. And
absolutely.
if
I
I
think
can
that's very
just
important. Go ahead,
I was
please.
going to say, if I can just add to this point, I think talking about all of the testing and validation before pushing to production is definitely something within life sciences, but it's also something that I continue to stress to all of the groups that I work with. We should move into some form of this, as opposed to just having a bunch of engineers sitting on the floor pushing live code to production. Vlad. But no, I guess having those multiple steps, having change control, having sign offs, having some sort of format of this is the way we go about doing it, as opposed to every day is just a completely insane day. At the end of week 3, our PLC or our SCADA or all of our code is going to look nothing like what it did at the end of week 1. I think there are too many facilities like that, and I think too many facilities get themselves really into the weeds by just continuing to push undocumented, untested, unvalidated code. So I would hope to see
Well,
more of a shift towards the larger structure that we were talking about.
absolutely. And what's really cool is the direction like things are going. So like something that I get excited about with Ignition is it's starting to become even more friendlier with
Mm-hmm.
Git. So in life sciences, like whether we like it or not, we got to document everything. And so, maybe like years ago, I think a lot of times what people do is they actually make literally Excel
Mm-hmm.
spreadsheets where they're saying, okay, here's my thing and here's all the parameters, and I update it by hand. And that's slow and painful and error prone. But what's cool is a UDT is just a JSON, right? So it lends itself very well to being documented in something like Git. So automatically, okay, I have a document of this is what the code looked like, and then so and so updated it. Wow, here's the difference.
Mm-hmm.
And I didn't have to spend much time to get that traceability.
Absolutely. I think that this is all super exciting, Amy. I think that we could go down like just an entire other episode and what like the manufacturing intelligence and data looks like. At some point, we will have an MI episode, and if it's you or someone else on life sciences, I absolutely want to go have a continued conversation on paper versus non-paper versus future MES solutions in the life science side. This has been an amazing conversation. We are getting close to time. Before we ask you the wrap up questions, I want to ask you maybe some takeaways from Operation Warp Speed, maybe things that were done correctly, maybe things that you would do different now. Kind of go to wrap up what could be like 7 hours of conversation. And just so all the listeners, I think we've talked to Amy about this for like 3 hours already off stream before we've come to this, and Vlad and I still have all of these other questions and thoughts. So kind of takeaways, things you did right, things that you would change in the future, please.
Yeah, absolutely. So as we kind of talked about, I think things that went really well with that project, and actually we're out of time, I didn't get to talk about it. The regulation and the flexibility of the FDA to adapt to the crisis was wonderful, because what we realized is that you can actually do things differently while still being compliant. And I think that set a really good precedent that moving forward, there's definitely going to be ways that we should probably think about doing things differently, maybe using more technology to help with that documentation aspect and validation aspect. So that was really, really cool. The other thing that I think they did well was they were very good everybody was really good about, again, having the right mindset. Because I think, oh gosh, sorry, I don't know why I'm choking. Having the right mindset of trying something different helps a lot. And I think that you can take that with you with any other project you do. That's going to set you up, again, really well to embrace new technology. Things that maybe I would do differently. My personal opinion is I prefer that people own their code. So honestly, that's not the state of industry. Everybody is buying these OEM skids. But my personal preference is long term. I think it's cooler, and you can probably do more, and you can get more insights if you have access to all of that plant floor control.
Interesting.
Sorry, that was
wow.
very quick.
There's a lot of different questions on that side, Amy. I'll be honest with you. I think that's all of those points merit their own discussion, and I know there's a lot of conversations in our industry around that, right? Like who owns the code specifically. But look, let me ask you a kind of like a slightly different follow up, right? And let Dave kind of close us with some final questions. But I'm curious, on the day to day on that project, right? Because again, for me, it was very structured and I sort of knew at least like 3 to 6 months what the schedule is going to look like. But for you, was it extremely chaotic and there were just constant changes, or was it very different? Could you paint us a picture?
Okay, no, it was.
I'm just like curious what it was like.
All right, here's. It was chaotic because what happened was all of the money in the life science industry was working on effectively the same exact project on the exact same timeline. So you had massive shortages of equipment and you have massive shortages of people. Because if you were about to hit your like installation qualification stage, so was everybody else. It was really hard to find engineers who could help support that. And then, okay, well, we need single-use bags. Yeah, so do you and everyone else. Normally, I think like the bag wait time was 4 to 6 weeks. At some point, it peaked to like 12-month lead times. And I was like, that's not going to work. So you kind of have to scramble and try to find, where can I find a single-use bag on the face of the planet in less than a year, And so not having like guaranteed equipment and guaranteed resources really impacted your schedule, because you had very finite structured, like, this is your run with cells. You have maybe 2 or 3 of these. Then this is your validation run. And all of that was so variable on what resources you had. So yeah, it was a little chaotic, but it was fun.
But so in that case, I'm assuming you're not the one who's purchasing the bags or looking for the bags, but you're shifted between projects as those things kind of happen, and so you're being pulled in different directions as I guess supply and demand.
Right. It's like, well, we needed to test the equipment, and it's hard to test the equipment if it's there or if it's missing parts. So you're like kind of bound, you have to be flexible and like, okay, we have a bag today, then we're going to focus on that. Okay, we don't, time to pivot to over here,
Interesting. I don't know how I would feel in such a chaotic environment. I'll be honest.
I was just thinking to myself, Vlad would not like that. I was just thinking to myself, that chaos is not something that Vlad would be happy with. Whereas I would go send someone to the grocery store to go buy me every size Ziploc bag that exists, and we'll see what gets close for some of this early testing. But no, this is awesome, Amy. We'll definitely continue to chat about this some more. But we do want to be respectful of your time, so we want to ask you all the questions we get to ask all of our guests. The first one, as you know, as all of our longtime listeners know, is super fun, is predict the future. And Amy, I mean, we've set ourselves up wide for what the future of life science is going to be. So maybe beyond you stockpiling single-use bags in your spare closet, can you predict the future of life sciences in the next 3 to 5 years, please?
Yes. Okay. So I think that we're definitely going to be using more process analytical technology, PAT. It's crazy how many decisions we make about processes with such little input. I don't think we've ever seen truly 2 identical trains, right? There's always something different. It could be the floor layout, and that does matter. So having more sensors will get you more information. I kind of mentioned it before. I think technology is going to make documentation better for life sciences. I'm hoping to see more PLC code and stuff that supports Git, so we can spend less time manually documenting like all the changes we made. And then the last thing is our industry honestly has probably spent the past 5 years starting to understand connect and collect and like the power of open architecture. So I'm hoping that 3 to 5 years from now, we're going to see a bit more on standardization. Okay. And the reason I say that is what's really cool is Jonathan Wise from SESME actually sent me a document which had authors from all the big names like Pfizer, Biogen, Cytiva, Merck, and they're talking about how are we going to do electronic data exchange on like shared data standards like ASTM E3077 for like raw material transfer. And there's like workshops now about how do we, make our supply chain more resilient. So I think the future is going to be a much more, hopefully, digital, resilient supply chain, so we don't have to stockpile all the single-use bags.
Very interesting.
I love that. Thank you for bringing it back to single-use bags. I know what Vlad and I will be talking about for the rest of the week. Vlad will be like, or I'll be like to Vlad, so do you think we can corner the market in single-use bags? And then we'll be like, Amy, what do these single-use bags actually look like? And then we'll go attempt to corner the market in single-use bags. But no, I think that's interesting. I've had several conversations with life science folks about the digitization process, about the digital sign-offs, about how some of the end users maybe had a less than great taste in their mouths because that was the leading bleeding edge concept of 5-ish years ago or 7-ish years ago. And then the implementation wasn't nearly as straightforward as we're getting off of paper sign-offs to digital sign-offs. So I agree that is the way industry is going to go for a bunch of reasons and has to go. And so I'm excited to see what happens with that on the life sciences side. Talking about life sciences, we like to ask for some career advice. And so typically someone kind of early career, maybe middle career, if they're looking for career advice specifically to get into life sciences, would like to know what your best career advice would be, please.
So if you're not in the life science area, honestly, the best thing is I would say any role you can that's close or adjacent to it. Because if I'm being honest, mostly what people look for on resumes is this knowledge of this GMP environment, because it is so different. You have to be so cautious of documenting everything. And then once you have that technical, like validated experience on your resume, it's a lot easier to then transfer roles into maybe what you actually want to do. But then if you are in the life science industry already, my biggest advice is talk to people. Because like I said, we do tend to have this culture of like information silos. So if you're in automation engineering, it's really easy to only know that, only do that, and never talk or meet the MES people, because like there's no short-term business justification for you to like have access to their system. But if you can talk to people, no matter who, especially if they're outside your group, you get such a better understanding of the process. And I think when you know the big picture, you can do your part of it much, much better.
Absolutely. No, I think that is fantastic. I think that is fantastic advice for anyone to go kind of step outside your silo, your comfort zone. I think it was a couple of weeks ago we had Glenn on. Glenn was talking about ERP, and that was a similar kind of piece of advice that he gave. Well, I mean, his advice is everything is financially model driven, so you just have to understand that nothing you're doing, unless you're on the financial side, is going to be the driver for lots of these decisions. But once you understand the bigger perspective, and once you're a little bit later in your career, you have a much better perspective of why some of these people have made the decisions they have, as opposed to my process needs single-use bags. Why can't we get more single-use bags, sort of thing. So thank you so much for that, Amy. Next question, we'd like to ask for a book and or content recommendation. What do you have for us today?
I like the book, The Technology Fallacy. It's actually a short read, but it's really good. It kind of highlights how most of your technology problems are more of like people problems, and it's more about becoming a digital company as opposed to just getting the latest tech.
Okay. I like that. Vlad, you'll have to. Vlad will go drop that in the chat and in the show notes for everyone who wants to listen. I feel like we've had that conversation. We might have gotten that book at some point in the past, but it's been a long time since we have had that recommended. So thank you so much for that, Amy. And last question for you is, how can our listeners help? Who should reach out and connect with you? What sort of conversations are you looking to have in your professional life at Skellig? Are you guys looking for more life science customers? Are you looking to hire? Very much kind of your open platform to ask our listeners anything.
Yeah, absolutely. So I think we might be hiring, was it North Carolina? Check our LinkedIn for that. We always post if we're hiring there. But yeah, we're always looking for life science clients who are looking to kind of embark on their digital transformation journey. That's a buzzword. I hate that. So we support, like I said earlier, like feed studies, architectural designs, automation, MES. But also, if you are a life science manufacturer and you feel like, wow, my site doesn't get it, we've been doing the same thing the same way forever, and you don't know where to start, reach out, because our company also does like workshops and educational presentations just to try to help people in our industry like make that change and course correct. So
Absolutely. Thank you so much for that, Amy, and everyone. Thank you so much for hanging out and chatting. We've had a couple of dozen people watching us live almost the entire time. Some people come on, some people drop off. We've had a bunch of good comments. Everyone loves you, Amy. I know that's not a surprise for anyone, but if you guys have made it this far, thank you so much for coming and hanging out on Manufacturing Hub, listening to us ask Amy probably very simple questions about life sciences. So thank you for all of our life science folks in the comments who are willing to let Vlad and I go ask some of those questions as you all probably rolled your eyes. If you're not already connected with Amy, please go make sure to go connect with Amy so you can have all of those fun conversations. Make sure you're following Manufacturing Hub and Vlad and myself. We are live every Wednesday at about 4 o'clock East Coast time. If you made it this far on YouTube or you're listening on podcast format, you're absolutely awesome. Thank you guys so much. Make sure to hit the subscribe button and hit the thumbs up button. Rate us 5 stars on your podcast platforms. And get ready, because when I remember to tell people to like and subscribe on the podcast side, and Vlad publishes our episodes on Thursday morning, more people listen, which is pretty awesome. Until next week, we'll see everyone soon.